Hello everyone, welcome to this webinar as part of our Shimadzu Solutions for Pharma series. I'm Brian Demansky, Strategic Collaboration Scientist at Shimadzu Scientific Instruments. I'll act as your moderator today, and we'll be hearing from our main speaker, Stephen Kurzniak, about mass spectrometry-based evaluation of biologic therapeutics. Before we start, I want to share a couple of notes for our viewers. The webinar console has a variety of items to help enhance your experience and interaction with us. In the screen, you'll see the following items. The slides will appear in the center of the console. Directly under the slides, you will see the related content list with clickable links relevant to the material being presented today. On the top left of the slides is the Ask Question widget. Please submit your questions during the presentation through this widget, and we will answer them during the Q&A session. Just below the Q&A box are survey questions that you may fill out anytime during or after the presentation. On the right are the moderator and speaker bios. You can expand these items to learn more about us. Finally, at the bottom of the pane are the icons to bring up any or all of these widgets in case they are minimized or hidden during the presentation. So let's get started. If you're just joining us, again, I'm Brian, your moderator, and we'll be hearing today from Stephen Kurzniak. In his role as a business development specialist for Shimadzu, Stephen uses his extensive experience in pharmaceutical method development and implementation to provide solutions for bioanalytical challenges. With that, I'll hand it over to Stephen.
Thank you, Brian, for the kind introduction. And as stated today, I will be presenting mass spec approaches to supporting biological development. Over the last decade, the pharmaceutical industry has experienced a shifting landscape. Where small molecule therapies once dominated, the biological-based drugs are now moving up. As of 2019, worldwide drug sales have pushed biologics to be 7 of the top 10, with 5 of them even being more specifically monoclonal antibodies. Each year, biological application approvals continue to rise, making this landscape even more pushed in the direction of biologics. Along with the shift from small molecules to biologics, the biological therapy landscape itself has become more diverse. And even though more focus was placed on protein-based modalities initially, mRNA and cell-based therapies have gained a lot of attention in recent years, especially with the cancer-based CAR-T approvals, and more recently a lot of attention from COVID-19 mRNA vaccines. But for today's presentation, I will keep the focus on monoclonal antibody workflows. Today's topics will focus on mass spec-based tools and workflows that can aid and better guide process development and ensure biologic quality with in-depth characterization of monoclonal antibodies. Over the long timeline for developing a new monoclonal antibody, a myriad of tools and methods are required to support research and development, clinical trials, bioprocess development, and quality control of the final product. But here is the good news. Shimadzu provides one of the most comprehensive portfolio of products and solutions, as well as software packages, to support that biological biopharma characterization. We offer such instruments as multi-TOF, triple quad mass spectrometers, ultra-high resolution liquid chromatography systems, modular as well as integrated systems for ease of use, as well as spectroscopy products, GC, GCMS, ICP, and ICP-MS systems, as well as TOC analyzers to support routine QC of raw material testing and applications such as cleaning validations. This wide portfolio of instruments can allow Shimadzu to be your analytical partner from concept to completion. Moving on, I'd like to provide a basic background for the structure of a typical monoclonal antibody therapeutic. The monoclonal antibody is typically divided into 2 regions, the FAB region and the FC region. The FAB region contains the variable sequence for the amino acids that would actually create a unique binding capability for the antigen or target component. It is this variable sequence in the region in orange that's highlighted that also is known as the CDR, and containing CDRs 1, 2, and 3, CDR stands for complementary determining region. The other component is the FC region, which contains a more constant amino acid sequence region. And in this constant region, it is the defining IgG subclass. And it is this region that helps mediate cellular effector function. The intact monoclonal antibody is actually made up of 4 individual proteins held together by disulfide bonds. 2 of the 4 proteins are commonly referred to as heavy chains. These are 50 kilodalton protein. And I have outlined it here in red in the structure of the monoclonal antibody. The unique feature of the heavy chain is that it actually contains a glycan modifier region. This is the small circle here at towards the base or C terminal of the heavy chain. The counterpart to the heavy chain is the light chain. And the light chain is a 25 kilodalton protein. And it is identified in this picture, outlined in red. Both the heavy chain and the light chain contain a variable region and a constant region. The variable region is identified with the color of orange, and the constant region is identified with the color of green. While the monoclonal antibody is made up of 4 proteins, it is made up of hundreds of amino acids. And those amino acids contain thousands of atoms. This chemical composition is created even more diverse due to the biological process of manufacturing monoclonal antibodies using cells. And these cells, during that process, will add multiple modifications to individual monoclonal antibodies, creating even more diversity at an exponential scale, which then would rival even the typical commercial airliner complexity. Because of the chemical diversity created by a biological process involved in creating monoclonal antibodies in a cell-based environment, monoclonal antibody development typically involves a stage called process development. During this stage, cell culture conditions are evaluated and determined to be finding the optimum conditions for both product quality and yield. And these conditions, done at a smaller scale, are then translated to manufacturing to produce a larger scale amount of product, which then requires a level of quality assurance and quality control. Over the next few slides, we will talk about how the LCMS 8060 can be used during this process development to monitor the cell culture process. The production of biopharmaceuticals, such as monoclonal antibodies, is a long and complex process. On this slide is outlined that complex process. Starting from the left, individual cells are added to a culture to expand and then placed in larger and larger bioreactors until enough cells have been produced in order to yield enough monoclonal antibody that is desired. Over this process, 2 key resources are spent: time and money. An unoptimized and unsuccessful bioreaction or production can waste both time and money, versus a successful one will keep the cost of both time and money to a minimum. The goal of the process development stage is to improve quality and reduce production costs and turnaround time. And how is that achieved? Through a better understanding of the process. At each stage, even early as evaluating the hybridoma, testing can be done for verifying product quality and product starting materials. Then moving on to cell culture, different monitoring conditions can be added and monitored for product consistency. And then looking at the final product, QC testing can be used to verify product quality. Now enter Shimadzu with a deep dive into cell culture profiling to better understand, to improve product quality reducing that production cost and time to making sure that successful runs every time. To help in understanding the complexity of the process development stage, Shimadzu has created the LCMS cell culture media profiling method. Using media in a single analysis, taking only 17 minutes, 125 media components can be monitored. This comprehensive list of components includes such things as amino acids and their derivatives, nucleic acids, sugars, vitamins, and metabolic products. In addition to the complexity brought on by the wide range of cell culture media components, the concentration of these components is also a wide range, going as low as 1 millimolar to up as high as 1 molar. While traditional methods may require multiple dilutions in order to achieve the appropriate detection range, the Shimadzu cell culture media method has been optimized to do a single dilution step so that all components are in the range for detection. The cell culture media profiling method takes advantage of the Shimadzu triple quad features such as fast MRM and rapid polarity switching. With polarity switching as low as 5 milliseconds, positive and negative ion components can be added to the same method so that all compounds can be analyzed in a single injection of 17-minute cycle times. The basic sample preparation for the cell culture media profiling method is outlined on this slide. As you can see that volumes such as low as 50 microliters can be used from the sample media with adding just basic solvents such as water and acetonitrile in addition to a small amount of a single internal standard. And the process largely involves mixing, centrifugation, and then a final dilution, which is then optimized by the end user based on their specific cell culture media type before injecting into the mass spec. As an initial and simple example of how the Shimadzu LCMS cell culture media profiling method can be used, here we analyze 3 different lots of fetal bovine serum. Fetal bovine serum is often added to cell culture media to stimulate cell growth. But variations, or lot-to-lot variations more specifically, in the fetal bovine serum could lead to variations in the cell culture media growth as well as in the final product, for examples of monoclonal antibody production. In this case, we've detected different components in the amounts and variations of products that were evaluated. And for these 3 lots, 56 different components were detected. Of the 56 components found in the fetal bovine serum lots, 10 components were found to have significant differences. And of those 10 components, 6 were nucleic acid-related compounds. With this method, one could analyze fetal bovine serum as a pre-screen and then match that to historically successful fetal bovine serum lots to ensure successful cultivation and product yield. In addition to lot-to-lot variation detection, the cell culture media profiling method can also be used to detect time courses to monitor cultivation from start to finish. In this example, we look at components that were consumed well within 16 hours. Once these components are depleted, they are not available for cells to produce or modify proteins. In the middle component window, we see that there are some components that were consumed over longer than 16 hours. And then also you can see secreted products for metabolitic components. As a tool in time course trend monitoring for cell culture, Shimadzu has developed the C2 Map Trend software. This software allows data from the cell culture media profiling method to be easily imported, allowing time courses to be visualized. In addition, multiple trends, multiple batches can be overlaid in order to see variation between various time courses and to evaluate whether or not different batch conditions may have changed the metabolic or consumption of different components. This is a published example of the Shimadzu LCMS cell culture media profiling method used in a specific biopharmaceutical setting. In this case, four batch-fed bioreactors were studied to monitor variations between different batch-fed conditions. The components were monitored from day 0 to day 18, and this was using CHO cell lines producing IgG subtype 1. During their observation of the four batch-fed reactors, they noticed components that, starting from the top left, increased over time, and the bottom left depleted early, the top right decreased over time, and the bottom right differences between batch-fed strategies. This paper also shared results comparing different methodologies for identifying lactic acid and other amino acids using different technologies and comparing them to the Shimadzu LCMS cell culture media profiling method, showing that the correlation between the orthogonal methods was consistent. In addition, in this paper, they identified an example of how the cell culture media method was used to help them solve a sequence variance problem. In this example, they identified in their final product that in a particular sequence, they found that a proline had been substituted with an alanine. When analyzing the cell culture media data, they found in their original feed that proline concentration dropped off fairly early in an 18-day process. Going back and looking at it, they then identified the fact that if this proline was then substituted for an alanine, that they would increase the amount of proline in the cell culture media at a particular stage. And then they found that by doing that, they actually then, the modified feed, then reverted the sequence variant from the alanine back to the proline, improving their product yield. So this is an example of how cell culture media would help to identify a solution and being able to correlate different data between the peptide level and the cell culture level. So the last example was a great way to transition to our next segment, which is the biopharmaceutical characterization segment. Now switching from LCMS triple quad to high-res mass spec using the Shimadzu QTOF LCMS 9030. Now we will look at instead of looking at the cell culture media analysis, now we'll be looking at the product quality at each stage, both during the process development and manufacturing, where the monoclonal antibody needs to be characterized at various levels. In this next section, we'll show how the Shimadzu QTOF LCMS 9030 uses high-resolution accurate mass spectrometry with sensitivity and speed to analyze the product quality of the typical monoclonal antibody. In addition to utilizing the Shimadzu LCMS 9030 hardware, we'll also utilize the Protein Metrics BIOS software platform for analyzing the protein data for the monoclonal antibody. The Protein Metrics BIOS software offers easy import of the Shimadzu LCMS 9030 data into any of the predetermined or pre-established workflows, which allows any user to quickly and easily process their data. Combining the high-resolution mass spec hardware of the Shimadzu LCMS 9030 and the Protein Metrics BIOS platform, you can see how easily we can support the analytical needs of the biologics R&D process. In this first analytical example, we use the NIST-MAB reference material to show how the 9030 and the Protein Metrics software can be used to analyze an intact monoclonal antibody. Because the 9030 uses electrospray ionization, the intact molecular weight of the monoclonal antibody of 150 kilodaltons actually receives multiple charges, which then creates a charge envelope. In this example, we see how multiple charge species occur over a wide mass range. But then in a zoomed-in feature, we see how each charge envelope area actually has multiple components in here. And these multiple peaks represent the fact that there's diversity in a glycosylation, which is something we identified earlier when we were defining what a monoclonal antibody structure was. After acquiring the raw data on the intact NIST-MAB on the 9030, we can import that data into the Protein Metrics BIOS intact workflow. This workflow allows the deconvolution of the multi-charge species spectra into a deconvoluted neutral format. And here we're actually able to then label different components based off of predetermined glycosylation modification masses. Here is an additional example of an intact mass spectrum for a monoclonal antibody known as Avastin or bevacizumab. And you can see that the glycosylation pattern compared to the NIST-MAB is different. And here's the NIST-MAB and the Avastin or bevacizumab side by side to show how the glycosylation patterns are different, having similar glycosylations but different abundances. After completing the intact analysis, now we move on to the subunit analysis. And the subunit analysis is now looking at the individual 4 proteins. And we look at that by doing a reduction of the disulfide bonds using different reducing agents such as DTT. Once we've done that, we will be able to look at the intact, the heavy chain, which will be around 50 kilodaltons, and the light chain. And here is the example of the light chain and heavy chain being separated chromatographically in the bottom chromatogram. And you see the spectra of the neutral form, so the deconvoluted forms, for each of the heavy chain and light chain, the light chain on the left and the heavy chain on the right. And one thing you can notice is that the light chain, which does not have the glycosylation region, has very limited or minimal heterogeneity, so it's largely one peak. Versus the heavy chain does contain the glycosylation region, so it has a similar heterogeneity, or at least some similarity to heterogeneity to the intact monoclonal antibody. Also analyzing the Avastin or bevacizumab, we look at the heavy chain and light chain. Again, the bottom shows the LCMS chromatogram separation for the light chain and the heavy chain. And on the left is the light chain, and on the right upper is the heavy chain deconvoluted spectra. In this case, again, we see that there's minimal heterogeneity on the light chain, and we see similar glycosylation presence in the heavy chain, since it does contain the glycosylated modified region. As we move on, now we'll focus on the deglycosylation of the monoclonal antibody to show how we can reduce the heterogeneity of the heavy chain by removing the glycans from its sequence. As we noted previously, when we looked at the heavy chain with the glycosylations on there, we saw a pattern that was fairly heterogeneous. now after we've removed the glycans using the PGNase F, we have found that the heavy chain now has been deconvoluted into a much simpler pattern, both for the NISMAB and on the left and the Avastin on the right. In addition to reducing the complexity or the heterogeneity of the heavy chain, the released glycans can actually analyze themselves. After using the enzyme PGNase F, which targets N-glycans, we can actually label the released glycans. In this case, we've labeled with procainamide, and this procainamide label increases both the UV absorption and the ionization efficiency of the labeled glycans for better analysis both on UV and mass spec. In this case, we have both the Avastin and the NISMAB glycan profiles, and so we can clearly see the released glycans and the structures that have been associated with the masses of the peaks. Our next stage of analysis now moves from looking at a subunit analysis down to a peptide-level analysis. So here we will be using trypsin, which is a protease that will digest proteins and will clip the protein at lysines and arginines. With foreknowledge of the NISMAB amino acid sequence, we can predict where the digestion cuts will be with the trypsin by identifying the arginines labeled R and the lysines labeled K in the sequence. So you can see clearly that there are plenty of R's and K's throughout the sequence. So we will end up with small segments or small peptides that we can analyze. Some may be larger than optimum, and then some may be too small to analyze. But we'll have plenty of coverage to be able to identify key portions of the sequence. If we were missing key parts of a sequence, we could always do different enzymes that have different cut sites in order to provide overlapping sequence covers for areas that might be too small or too large. After performing our tryptic digest on our proteins, we now have a pool of peptides, which we have run here on a LCMS chromatographic separation. So now here we see there are peaks of the different peptides, and on top we have the NISMAB, and the bottom we have Avastin. You can see they're similar, but obviously they have differences because different sequences will provide different peptide cuts. But in addition to having chromatic separation, because we have the mass spec, we will have the intact mass to know what the full peptide sequence is, as we will have MS data, so then we can confirm the sequence. With the acquired peptide digest chromatographic data, we now have intact mass as well as MS data for each of the peptides in the chromatogram. We import that into the post-translational modification module within the bio software, and then we have our pre-understood sequence information from the NISMAB, both heavy chain and light chain, and that information now shows the sequence information as the single letter codes, but we have lines here indicating what peptides were identified. You can see from those cases that we have peptide coverage across the entire sequence, and sometimes we have different size lengths because sometimes the sequence does not get fully digested, so you have miscleavages, which means that there may be multiple peptides that were actually created to create a larger peptide. But you can also see that there are different colors in here sometimes, and that indicates modifications compared to the green, maybe the orange. It also points out here in the yellow boxes the CDR regions, so that shows where the variable sites are for the interaction with the antigen. So we want to make sure in many cases that we have that coverage because that's the variable region identifying the uniqueness of the monoclonal antibody. And again, as an additional example, compared to the nismab, we also have the Avastin or bevacizumab monoclonal antibody. And here we also see the light chain and heavy chain have coverage. And here is an example of how one can monitor modifications that may vary on one monoclonal antibody to another within the same batch. The individual monoclonal antibodies may have what is the expected wild-type peptide versus a modification to a peptide of the same sequence. And this may occur during the process or actually during storage. But in both cases, when you digest down to a peptide level, you'll be able to compare the two different sequences that have the similar base sequence but a modification to it. And Proteimetrics software does a very good job at being able to easily compare those two and identifying those two very quickly, and then also providing a relative modification percent, so that we can see here that 13.22% of the relative amount of total peptide is deamidated. And this is also brings back our example of how we compare it in the cell culture, how we were able to take the same data and be able to identify something like this and maybe relate that back to bioprocess information. So now we're coming full circle and talking about how we use both the cell culture to track down maybe what happened, but then here we're identifying where the modification occurred. So, and this can be used as a critical quality attribute in a multiple attribute monitoring methods, so which means that once you identify that this is a potential modification that could occur that maybe is not desirable and maybe has some efficacy issues or stability indication issues, we can then monitor that more specifically and create a method dedicated to this. Now, after going through the two different segments of our talk, focusing on the cell culture media analysis with the LCMS 8060 and the C2 map trend software, and the protein monoclonal antibody characterization, set with the LCMS 9030 and the Proteimetrics BIOS software platform, we can see definitely how our solutions can help you monitor and optimize your bioreactor and cultivation process for process development, and then also monitor and make sure you have consistent high-quality biopharmaceutical products like the monoclonal antibody. Just like the complementary components of our hardware and software for the different workflows, I'd like to acknowledge the complementary people that helped me with generating data for this presentation. So I'd like to put a special shout out to Vicki Johnson, who did a lot of work on this, Chris Gillis, who is our product manager for the mass spec product lines, and Evelyn Wang, who is an application scientist at Shimadzu Scientific, as well as our partners at Proteimetrics, Sinjin Skilton, and Michelle English, who have been great ongoing support with making sure that Proteimetrics integrates properly and efficiently with our data. Thank you. And I'd like to thank everybody for your attention today, and I hope that we have provided some valuable information. And I can be reached at my email address at stevenjkersnick@shimadzu.com. For any additional questions you have, please reach out to me. You can also visit our vanity URL, which is solutionsforpharma.com, for this information, as well as for more information about other workflows, as well as up-and-coming events. Thanks for your attention. And now we have time for questions.
So let's again thank Steve Kersniak for that engaging presentation. As you can see underneath that related content list, there's more information about our analytical solutions, including the full application notes regarding Steve and Vicki's work to create that complete characterization workflow for NIST-MAB that you saw portions of. So if you want more detail on the methods, the instruments used, and the approaches, you can find that information in that related content list. There's also some detailed brochures that have a wide range of solutions that Shimadzu provides for biologic and bioanalytical development. If you haven't had the chance, we ask that you fill out the survey questions on that left-hand side as well to give us any feedback or request any additional information you may want from us. We encourage you and your colleagues as well to sign up for the Shimadzu e-newsletter by responding to survey question number 3. That monthly newsletter includes information regarding upcoming webinars and events that encompass a wide application area range beyond that of pharma as well. Obviously, we have a number of analytical solutions that can be used across a number of different markets. And we'll go ahead and begin our Q&A session at this time. We encourage you, if you haven't yet, to submit any questions that you may have through the question widget in that top left corner. And we'll go ahead and take a look at some of the questions that have come in already. So the first question that we have is, are the workflows that have been demonstrated able to be adapted for GMP and GLP compliance?
That's a great question, Brian. Yes, actually, in most cases, or pretty much all the cases, these can be adapted. I would say definitely the proteomics or the protein characterization is definitely something that's already been pushed in that direction. On the cell culture, which is typically monitored a little bit earlier stage, it's not necessarily typically done, but you can, because it's a small molecule, create quantitative methods eventually using that. So yes, those can be adapted to GLP, GMP workflows.
Excellent. Do we have software that can support that?
So the software with lab solutions could be able to support that, as well as the protein metric software can support that environment.
Excellent, excellent. We have another question related to that protein metric software. There was a question regarding N-glycan analysis. And are you able to use that software to incorporate LC fluorescence data along with MS?
That's another good question. Yes, that's something that we did demonstrate, and it is available in the protein metric software to integrate those alternate detectors in addition to the mass spec data, such as fluorescence and UV.
Excellent, excellent. It seems like that's a popular topic, is the software as well. It had been mentioned in the presentation that there had been a trypsin digest done, and the question that came in is asking whether or not the software is compatible with digestion workflows other than trypsin for peptide mapping.
Yeah, and that is definitely something that's probably very common amongst different companies where they're trying to get a complete coverage of the monoclonal antibody. Because again, we saw that sometimes there are small sections that don't get coverage because they're just too small when you do a digest with trypsin. But yes, you can use additional, like lysine C or leucine C, and all these can be actually added together, so which means you can create 3 different samples, combine the data together to get 100% coverage. In addition, which was not mentioned here, but you can again find it at solutionsforpharma.com, is an automated system which does online digestion, which is called our Perfinity system, and that can be added to the front of a QTOF to basically automatically digest and then send on the digested sample onto a column for separation and for the mass spec for detection. So that's a great way to add, and that can add multiple digestion columns as well.
Excellent, excellent. Thank you for that and wonderful information. Looks like we have at least one other question that I can see at this point. It's asking, at the end of the presentation, you had mentioned MAM, multi-attribute monitoring methods. Are these currently accepted for use in initial filing with the FDA or final product analysis?
Yeah, and actually this is an ongoing kind of consortium kind of thing of making that whole process easier. And so the MAM, and the multi-attribute monitoring, is a way to basically say, hey, we know these peptides are unique characteristics of the monoclonal antibody are typically expected to be a particular peptide, but then modifications can occur. So if we know what the modification is, we can then predict or create a method that can monitor both the modified version and the wild type, so that we can determine quick percentages. And then instead of having to do something that's always discovery, we have a very defined plan. And yes, and those things are now more readily implemented into regulatory processes and part of the package for the FDA to review.
Yeah, and I'll add to your answer there, Steve. I know from some of the work that I've done that the Center for Drug Research and Evaluation, CDER, a branch of the FDA, is trying to incorporate more of these LCMS-based methods both into drug filings as well as then that final product analysis phase. So there's a movement within industry and amongst regulation and regulatory bodies around the world for these biologic therapeutics to have that greater level of characterization. So that's certainly something that we see growing in impact and creating a need for these types of solutions, including especially that high-res mass spec portion. So unfortunately, it does look like we've reached the end of our allocated time for today. So if you do have any other questions, I would encourage you to submit those through the question widget. We will answer those offline. Anyone who has submitted questions up to now will also respond to you directly via email if your question was not answered here in this format. So we thank you for all of these wonderful questions. If we don't reach out to you now, we will certainly send you that email contact. Once again, thank you all for attending and participating. We will send you the link with the recorded version of this presentation that you can view anytime on demand. And we again encourage you and your colleagues to attend our upcoming webinars. We'll have registration information that will be sent out shortly regarding our upcoming series on biologic therapeutics, as well as other opportunities. And you can find any upcoming webinar events at our website, solutionsforpharma.com. Thank you and have a great day.